I must give the British people a very simple instruction. You must stay at home.
Many companies have now shut down, whether it's bars or restaurants.
I know that people are worried about losing their jobs.
We're looking at sending checks to Americans immediately.
For the first time in our history, the government is going to step in and help to pay people's wages.
The coronavirus pandemic is making it even more imperative that we introduce a basic income as quickly as possible in every part of the world.
Calling on the federal government to immediately begin a universal basic payment.
Will the Prime Minister stand up today and give a commitment to provide people with the security of a universal basic income?
100 years ago exactly, there was the Spanish flu, which was a global pandemic by which over 40 million people across the world lost their lives. But what was extraordinary about that was that there wasn't an economic crisis. By contrast, the coronavirus pandemic is going to be associated with an incredible economic crisis that spreads across the world. And it's this combination of the pandemic and an economic crisis that is going to cause many, many more people to die or have lives of misery. Since globalization began, the economic system of the world has become increasingly fragile, fragmented, and dangerously unbalanced. In addition, we have the biggest crisis of all, and that is the ecological crisis, the fear of extinction, the realization that we have to change our growth model, that we have to have more sustainable respect for the environment and the way we live. It will become a legitimate part of our social fabric very quickly.
The outer surface of a cell is a very complex surface, lots of little bits and pieces of molecules and compounds sticking out in all directions. And the immune system can recognize these epitopes, these little projections, as cellular ID tags and uses them to separate self from non-self. Both H and N spikes act as antigens, as do two proteins that are inside the core of the virus. And they can be readily identified by the immune system and are diagnostic. Small changes in antigen structure are called antigenic drift, and they create very similar strains called variants. Larger changes, called antigenic shifts, create different strains called subtypes. These new subtypes that are responsible for major new outbreaks of flu. When the virus attacks, it sticks to the outside of a cell by its H spikes. The spikes lock onto a type of sialic acid sugar that's found on the outside of cells in the lungs and in the throat. And they repeatedly bind to those sialic acid receptors like a Velcro strip or like tiny little pirate grappling hooks. It is specifically designed by evolution to cling to a molecule that protrudes from cells in the throat and in the lungs. So here's the virus now stuck onto the outside of a cell. It's absorbed into the cell through a process of phagocytosis, in which small particles can literally be surrounded by the cell membrane, drawn inside, leaving the particle wrapped in a tiny little bubble of cell membrane called a vesicle. Many other kinds of viruses fuse themselves to the surface of the cell in order to inject their contents, but this strategy leaves them open to discovery by the immune system. By slipping inside the cell membrane intact, and by wearing the cell membrane like a wolf in sheep's clothing, the flu virus makes itself invisible to the many wandering immune system cells that are scouting the body for trouble. Once primed inside the cell, the virus sheds its envelope and releases its RNA genes. Now, what we refer to as a gene is simply a series of coded instructions along a length of DNA or RNA that codes for putting together a particular protein. And proteins are the building blocks of life. Each strand of RNA or DNA consists of a long series of genes which are basically recipes for proteins. Each separate gene is a separate recipe. A chromosome is really just a series of genes on a single very long strand of DNA, all coiled up into a tiny little package. Now, viruses contain relatively few genes, and these are usually on a single strand of RNA or DNA. But the flu virus has multiple strands of RNA, each with only one or two genes. The RNA genes of influenza, once primed and liberated in the cell, hijack the cellular factory, replacing some of the host cell's genes and reprogramming the cell to make thousands and thousands and thousands of copies of the flu virus. The flu genes replicate and group together in sets of 8 to form a new core, and each set of 8 is wrapped up in a fresh membrane, and then they exit the cell to spread the infection. Within 10 hours of infection, that cell can release 100,000 to 1,000,000 or more new flu viruses. Theoretically, new viruses should become trapped as soon as they exit the cell by the same sialic acid receptors that they stuck to on the way in. That's where the N spikes, the neuraminidase spikes, come into play. They have a blunt tip. It's kind of like a little box with 4 miniature propeller blades. And these little blades slice through the receptor sites as the virus emerges from the cell and prevent it from being stuck. A mutation is just a change in genetic information, a change in the recipe that determines how proteins are put together. And even a small change in the exterior antigens of a microbe can cause the immune system to fail to recognize it anymore. And the influenza virus has an extremely high rate of mutation, makes it a real genetic chameleon from the standpoint of the immune system. Now, many mutations are stopped in their tracks because DNA has a built-in proofreading mechanism. So every time DNA is replicated for cell division, it's very carefully checked to make sure everything is as it should be. But RNA lacks that proofreading mechanism. So mutations of RNA aren't repaired, they aren't eliminated, and that means that an RNA virus, like the flu, has a much higher mutation rate than a DNA virus, thousands and thousands of times higher. We call it hypermutability. And that means as many as 99% of the newly created flu viruses are basically damaged. It also means that between 1,000 to 10,000 viruses from each infected cell can not only still infect other cells, but may now be even more lethal than it was before and better able to hide from the immune system. Mutations are thought to be the explanation for antigenic drift, small changes that create new variant forms, like the change that caused the second wave of the 1918 flu to be so virulent, could result from the fusion of 2 different types of flu viruses, from hybridization. But the way the flu virus reproduces actually makes it relatively easy. Remember that the 8 genes of the virus are on separate strands of RNA. So if 2 subtypes, if 2 variants, infect the same cell at the same time, each is making thousands of copies of its own 8 genes, which are all now scrambled up together inside the same cell. And when the genes are reassembled in groups of 8 to form a new core and sealed into a fresh viral envelope, the 2 types can very easily mix together. So the new virus will still have all 8 genes that it needs to survive, but those 8 genes are now going to be a random mixture of the genes of both types of the flu virus. It's only by reconstructing the virus that we can determine its origin and test this hybridization hypothesis about its virulence. Well, the virus could also have strengthened simply through passage from host to host. It's easy to demonstrate in the lab that as a new virus infects new victims, it somehow seems initially to strengthen with each passage, something we don't really understand. It may be that each new passage provides a fresh opportunity for mutation or hybridization. We really don't know.
And as it does so, I think you will find people having a greater sense of freedom, a greater sense of community, and will want to shift their lifestyles to spending more time doing work that is not labor, doing care work, doing community work, doing voluntary work, doing ecological work, doing the sort of activities that don't eat up the resources and eat up the planet and cause extinction, but rather actually help to reproduce our communities, our sense of localism and our sense of slowing down. And that, I think, is a very healthy prospect, because then people will feel they're gaining control of their lives. And in that process, I believe new forms of social solidarity, new forms of collective action will spring up. And I think the potential for that is a wonderful liberation, a wonderful return to the values of the Enlightenment, the values of community, the values of creativity and leisure, real leisure in the sense of the ancient Greeks, looking after our culture, looking after the values, and reproducing the ethics of empathy and compassion